Welcome back to ACCEL for June 2012. Disc 3 begins with a conversation Bob Vogel had with Dr. Paul Franks, Professor of Genetic Epidemiology at Lund University Diabetes Center in Malmö, Sweden, and an adjunct professor at Harvard, on are genes a reason for obesity and their ills or just an excuse?
Welcome to ACCEL, Paul. Paul, tell us basically how genes and environment interact. Is it just, the old adage, genes load the rifle and the environment pulls the trigger?
So I think that's a really nice way of putting it. Essentially, we all have differing genetic predispositions to disease. Many of us live a similar lifestyle today where we don't do so much physical activity as our ancestors might have done, and we eat very different diets. And some people are clearly more predisposed to develop cardiovascular disease than other people. And at that level, that's probably what we're talking about when we think about gene-environment interactions.
So there are several good examples where we think environment has played a big role in the expression of genetic predisposition. The Pima Indians, for example, might come to mind.
Right, so the Pima's a nice place to start this story. They're a group of individuals that today have a terrible burden of diabetes, obesity, and other related complications.
But it certainly wasn't always so.
Right, so if you go back to really before 1910 when the Roosevelt Dam was built, then you'd find very little diabetes in Pimas before that. In fact, if you look at the photos of these guys, they were athletic, really fit-looking guys.
You bring up the question of obesity, and certainly the important question is how much does genetic basis load the obesity gun and how much does environment pull the trigger?
Oftentimes people talk about nature and nurture, really, which is more important. The reality is they work very much together. They're part of the same deal. So, we're exposed to our environment, whether it's an obesogenic environment or not, 24 hours a day. And our genes are the conduit for that signal to translate it into our health and disease phenotypes, the way in which we outwardly express the predisposition and the eventual endpoints for disease. Really, it's not a question of which is more important, it's how they interplay with each other. And today we live in an environment that's very different to how we evolved. We live in one where we're essentially encouraged to be physically inactive and to eat very calorific foods. And some people's genetic makeup doesn't really accommodate that. They weren't designed in that way throughout evolution, and so today environmental insults have a much bigger impact on a genetically predisposed person to obesity than somebody who isn't. And you can look across different ethnic groups to get a good handle on that. And we talked a little bit about Pima Indians a moment ago. Clearly they're a group that have been very affected by this transition that occurred in the early part of the 20th century in their lifestyles and developed a lot of obesity and diabetes, whereas people from Central Europe appear to be far less susceptible to those environmental insults, and that's probably underlying that a genetic factor.
How far have we come in identifying specific genes that give a propensity toward obesity?
So there's different ways in which I can answer that. Since 2006, really the point where the way in which we study human genetics at a population level changed massively with the development of something called a genome-wide scan. And after that point, we really made massive gains in understanding the specific regions of the genome that are likely to influence our predisposition to obesity. The clinical utility of that is very limited, that has to be said, but in terms of how we've developed in these last few years, we now know many, many more times more about the genetics of common obesity than we did prior to this transition in 2006. For rare forms of obesity, monogenic obesity, there are much more clinically impactful observations at a genetic level, but of course they occur at a very low frequency in the population, and we're really talking about two very different types of obesity and implications related to those two types of obesity.
So right now you cannot send out for a gene screen for your likelihood that your kid is going to be overweight?
Not so that it means much, no. I think nowadays we're really focusing on trying to determine at a population level what's reproducible and reliable in terms of genetic associations. And probably within the next 5 to 10 years, that will be filtered down to an individual level, and there may be tools available where you might genotype patients, for example, to understand their predisposition. But today, despite the availability of many direct-to-consumer genotyping kits over the counter, the actual value of those for complex diseases is minimal.
As it is for picking out coronary disease risk.
Correct.
You can also change the functioning of your genes through environment. It's not just eat a lot in someone who's susceptible to gaining weight. In fact, through control mechanisms, you can actually change how your genes operate. Could you talk a little bit about that?
Right. So, we can talk about genes at different levels. And at one level, we talk about nucleotide sequence variation, which is essentially the same from birth to death. You don't change your DNA sequence. But that doesn't mean that the way in which the genome expresses itself doesn't change. It does change, and it changes throughout life, a natural function of aging, but it also changes depending on what we eat, how physically active we are, how much sunlight we're exposed to. So the structure of the genome stays the same, but the way in which it behaves, if you will, changes depending on the environment. And we really want our genome to behave in a way that's most friendly to us. And that means putting ourselves into an environmental context that allows the genome to behave in that way. Now, when we talk about gene-environment interaction, what we're saying is that some people's genomes are probably far more resilient to harmful environments than others, and that those people that are resilient, well, they may get a fairly easy ride in this modern industrialized lifestyle. The people who have susceptibility variants, they're the people that we might want to try and pick out early on and help them avoid smoking, for example, or other harmful environmental factors, and put more resources into those people to prevent disease. But of course, we have to be at the point, first of all, where we've got reliable genetic information to help us do that.
Can you pass on the good or bad effects you've had on your genome to your children or grandkids?
So there's some really fascinating work, some of it that emerged in the north of Sweden, in fact, where multigenerational effects of famine have been observed. And many other studies from Holland, for example, that have shown that people who are exposed to famine in utero may, several generations later, have people within their family lineage that are influenced by that exposure. And those effects are so-called epigenetic effects, and they're thought to involve changes in the way in which areas of the genome are methylated, or ways in which the histones are changed, and some other epigenetic effects. But essentially, these transmissible changes in the way in which the genome behaves have certainly been observed in animal models and at the population level across generations. I think the very exciting and somewhat disturbing thing to think about over the next few years is essentially, does the way that I behave now, before I even have my children, impact the health of them and perhaps even their children in the future, the way, how physically active I am if I smoke and so forth. So there certainly appear to be these multi-generational changes that occur without actual structural changes to the human genome, these so-called epigenetic changes.
Is this a process of evolution?
So methylation is one of these epigenetic changes, is there for good reason, and it's to help cells express themselves in different ways and behave in different ways in different areas of the body. And we need that flexibility to survive in evolution. But the reality is that the way in which we live today is so vastly different, it's so separated from how we evolved as human beings. And throughout the vast majority of evolution, we probably had fairly low calorie dense diets. We probably had to be physically active to get that food. We were probably exposed to heat and cold to far more extremes. And we probably, some populations certainly would have migrated and had a great demand for physical activity through migration. And those factors may have selected out variations in the genome that are advantageous under this enormous physiological stress that was common throughout human evolution. Well, today we don't have those physiological stresses, yet we have a genome that's designed to function well with those types of environmental factors. And if you take those factors away, you now have a genome that really doesn't fit with the environment, and that is probably why we're developing diseases that are associated with energy metabolism today.
So the basic message is that we ought to give thought to how we were designed to live.
Correct.
Paul, it's been very nice having you with ACCEL and talking about some fascinating new material.
Thank you.